Good morning, everyone. My name is Soumitra Pal, and I am from the National Institutes of Health, USA. The title of my presentation is Stochastic Modeling of Gene Expression Evolution Uncovers Differences in Evolution of Species, Organ, and Sexes in Drosophila. Let us consider 9 Drosophila species which have significantly different phenotypes. For example, the top 6 species from Sophophora subgenus thrive on rotten fruits, while the bottom 3 from Drosophila subgenus thrive on rotten vegetables. One question that comes to our mind, can we identify genes responsible for adapting to different environments? Or in general, can we identify genes that evolve neutrally or in a constant way? To address this question, we collected RNA-seq data from 5 tissues from the flies of both sexes for these 9 species and in 4 replicates. Total 360 samples we collected. And we analyzed this data using a stochastic model named as Ornstein-Uhlenbeck, or in short OU process. In this model, let us consider a phylogenetic tree with 2 species I and J. The expression for these 2 species initially were same, but after some speciation event, the expression diverged. If XIT represents the gene expression at time t since root for species I, then the incremental change in expression for species I in time delta t can be computed using this equation, where the right hand side has one term due to Brownian motion which generates a random deviate, and the other term tries to revert back the value towards an optimum theta I, and also controlled by a non-negative parameter alpha. The expression value of replicate K of species I is assumed to follow a normal distribution where the mean is equal to the expression value of species I at time ti at the tip of the phylogenetic tree, and with a variance parameter gamma sigma square. Here we have shown one replicate value in using green color for species I and purple color for species J. If we stack the expression values of all replicates of all species, we get a long vector which can be shown to follow Gaussian distribution, and hence the parameters can be estimated from the data. Depending upon the values of parameter, we consider 3 scenarios. Alpha equal to 0, when only the Brownian motion is active, we call it neutral. In the general case, alpha greater than 0 is called constant. We also consider a special case when multiple optima is active in the theta. We call this case adaptive. Once we have figured out the 2 sets of genes, neutral and constant, we wanted to compare the evolution of these 2 sets of genes using a metric called divergence, which is equal to 1 minus Spearman's correlation coefficient of expression values of species I with respect to a reference species J, in this case Drosophila melanogaster, and plotted against the time between the 2 species. We then fitted Michaelis-Menten curve from the enzymatic kinetics theory, which has 2 parameters Vmax and K, which helps us quantitatively compare the expression evolution across tissues and sexes. In short, for the neutral, this curve is expected to be straighter and going up. For constant one, it is supposed to be flattened after some time towards an optimum. We compared this for all tissues, which are shown in different colors, and sexes. Males are shown in dotted and females in solid line. We found out that the male gonads are least constant, in the sense the maximum divergence value is way higher than other species and sexes. We wanted to see if the neutrally expressed gene can reconstruct the phylogenetic tree, which is given here. And when we computed the never joining tree from the female gonad expression values, the tree obtained is almost same. It has Robinson's foul distance 0. We did the same for all tissues and sexes and figured out that except for male head, the neutral genes gave the tree, which is at least as good as the tree often considering all genes, and in fact many cases the tree matched the sequence based tree. Next we used our tool EvoGeneX to find out adaptive genes. For this we considered the two subgenus Phocophora and Drosophila introduced earlier to have two different optimum values of gene expression. We applied the tool to expression values in male gonad or binding proteins, and our tool found out that OBP44A is significantly lowly expressed in Drosophila subgenus and OBP56H significantly highly expressed in Drosophila subgenus in compared to Phocophora. More results on the other tissues and sexes in the manuscript. In summary, OU process is a good useful tool for modeling gene expression evolution, and incorporating replicate data improves inference. The details of the code and the manuscripts are given here. I have a talk at the EvolCompGen COSI track on Tuesday, 14 July at 4:10 p.m. Thank you for your interest, and I'll be available for questions and suggestions in the chat sessions. Thank you.